Hello everyone, welcome back. Now that we've completed our discussion of cellular structures and how cells come together to form tissues, we're going to scale up and start discussing how tissues come together to form organs, and then organs form entire organ systems. The first organ system we are going to cover this semester is the integumentary system. We'll start with an overview of both the structure and the functions of the integument. The primary function of the integument is to act as a protective outer covering of the body, but it also acts as a protective lining on the inner surface of our hollow organs, such as our stomach or our intestines. The integument itself is composed of, or it's secreted from, epithelial tissues, which we talked about in our histology unit. Remember, epithelial tissues are made up of epithelial cells, and they include things like glandular tissue. Epithelial tissues line the insides of organs or cover the outer surfaces of the body, or any part of the body that's exposed to the outside environment, such as the surface of our tongue, our cheeks, or the lining of our throat. Okay. Remember that glandular tissue, it's a specialized kind of epithelial tissue, right? Frequently made up of simple cuboidal epithelium, and it's responsible for producing chemicals, things like saliva or sweat or oils. Integument has a lot of other functions though as well, in addition to being protective. It helps to prevent water loss. It seals moisture inside the body so that animals don't desiccate as easily. Right? Desiccation is simply drying out, losing water to the environment. Desiccation. That's a word you'll need to know. Integument is also responsible for more fun stuff, like displaying body coloration, like what you see on this bee here on the right. All of those color molecules are embedded in the exoskeleton of the insect, and that exoskeleton counts as its integument. And we'll talk about exoskeletal structure at the beginning of the second half of this lecture. Gas exchange also occurs through the integument in some organisms, especially marine or aquatic life that does a lot of exchanging of oxygen with the water that it lives in. Again, secretion of glandular compounds, things like sweat and oil, are secreted right onto the surface of a mammal's skin to help cool them down or to help waterproof them, seal in moisture, again, prevent desiccation. And finally, the integument acts as a supportive network for embedded nerves and blood vessels and sense organs. So the neurons that detect tactile senses, touch senses, they're embedded in the deeper layer of our epidermis. And that deeper layer, the deeper layer of our skin, which is called the dermis, it's actually under the epidermis. And that cushioning layer houses the nerves and provides structure and support for them. What you're going to see over the course of this lecture is that as animals diversified over evolutionary time, the integument changed form as those animals started to move into different environments. You'll see the biggest changes as animals moved from an aquatic environment to a terrestrial environment. And also, you're going to see changes in integumentary structures as animals began to increase in overall size. Right? You'll see evidence of increasing complexity of integumentary structures as animals had to adapt to new and variable conditions. And you're going to see a lot of big changes in response, especially to a terrestrial lifestyle. One of the biggest changes that animals had to endure when they moved out of the water was the drying effects of air. Losing water to your environment is one of the easiest ways to weaken yourself and to eventually die. So the prevention of desiccation was one of the biggest challenges that animals had to face when they moved out of the ocean and onto land. So overall, the specialization that we're going to see arise in different integumentary systems is in response to changes in size and changes in environmental conditions. We will start our discussion of integument with pseudo-integument, you could say. We're going to look at phylum Porifera first, which, as you'll recall, doesn't have any true tissues, which means technically they can't have integument, since integument is an organ made out of tissues. Instead, what they have is a layer of cells that acts as an early integumentary system. Always remember, in poriferans, their body covering isn't derived from epithelial tissue because they don't go through gastrulation, which means they don't develop tissue layers. Remember, poriferans have a cellular level of organization only, which means no true tissues in this group. Instead, what poriferans have is a protective outer layer of cells called the pinacoderm. Okay? Derm, this suffix, think of it as meaning skin. Whenever you see that suffix attached to a word, it tells you you're probably talking about integumentary structures. Now the pinacoderm is here. It's this outer layer of cells. Okay? So the outside environment is on the right of this slide. The inner environment of the sponge is on the left. Okay. You see our choanocytes here. Hopefully they're very familiar to you now. And these orange cells on the right make up that protective outer layer called the pinacoderm. Most of these orange cells that you see are called pinacocytes. They're thin and elastic, very stretchy, and they act a lot like simple squamous epithelial tissue in animals that have tissues. They make up the majority of the body covering of sponges, but in amongst those pinacocytes, you find more specialized cells that have different jobs. For example, you have myocytes. Myocytes are contractile pinacocytes. Contractile meaning they can squeeze shut. You find them surrounding the ostia and the osculum. They allow the ostia to close and open adaptively. So if the sponge needs to shut itself off from the surrounding water for a while, it can actually close all of its ostia and end the flow of water through its canal system until it's ready to open them up again. Same thing with the osculum. Okay. Now, the ostia themselves, those openings through which water flows into the sponge, they actually aren't gaps between cells. They're a cell in and of themselves. The ostia, those openings, are made up of cells called porocytes. So porocytes are these tunnel-shaped cells, like what you see here. This entire blue structure is a porocyte. It's shaped like a tube, and it allows water to flow into the sponge. The opening that the porocyte makes up is what we call an ostium. Now the pinacoderm overall is almost a tissue. It's really close to being a tissue. It's right there, except that developmental stage that gives rise to true tissues is missing in this group. If not for that, the pinacoderm would be a true tissue. But the cells within the pinacoderm work together so closely, they approximate the functions of an actual tissue. They come real close. This gives us insight into how tissues originally evolved. Cells came very, very close to forming true codependency in our ancestral sponges. It just took the development of that gastrulation stage in embryology to push us over the edge into true specialization, where cells are so codependent on each other they cannot live independently. That's what gives you true tissues. And that's what shows up in our more derived group, the radiata. Cnidarians and ctenophores, the ancestors to cnidarians and ctenophores, were the first organisms to evolve that had a true integument. Remember that the radiata are all radially symmetric, hence the name, so they've all got this circular symmetry. Remember too that they're the oldest extant diploblastic animals. Extant meaning they're still living. Diploblastic meaning they've got those two embryonic tissue layers, the endo and ectoderm. Okay. This means they've got true tissues, so what we're going to look at here is a true integumentary system. This integumentary system, though, is pretty straightforward. It's about what you would expect from the closest living relatives with tissues to our ancestor who lacked tissues. Remember, in Porifera, you had a whole bunch of cells working together in that panacoderm so that they really resembled a true layer of tissue. Here, you've got almost the exact same diversity of cells, only now they really are codependent on each other. Radiata tissues in the integument just take that next evolutionary step into true codependency. None of these cells can live independent of the others. In this integument, you have a single layer of epidermis. That's it. One layer, one cell thick. That's your entire epidermis, your entire protective body covering. It's made up of epithelial cells, as you'd expect, but they're spaced out to create room for another kind of specialized cell, called a nematocyst. Remember that nematocysts, these guys here, these are your stinging cells. They are what cnidarians use to hunt and capture food. Now those nematocysts don't just develop on their own. Remember, nematocysts get used up when a jellyfish hunts with them. Once they shoot off their harpoon, they're used up, they can't be reloaded, and they can't be recycled. Instead, they have to be replaced. So where do you get your replacement nematocysts? You get them from stem cells called nematoblasts. Nematoblasts are immature cells that eventually give rise to mature nematocysts. Okay. Here are your nematoblasts, here, in this part of the diagram. Each of these little guys is an immature nematocyst, just waiting to be released so that it can replace a used nematocyst. You have other cells and structures here as well. Not pictured on this diagram are pigment cells. These provide color, as you would expect. And also, there are sensory cells. You do have a sensory cell here, formerly called a sensory neuron because it's part of the nervous system. They're these hair-like protrusions which, when pushed on, when this hair is contacted, send an electrical impulse to the central nervous system of this cnidarian, telling them they've been touched by something. You also have gland cells. Gland cells do exactly what you would think. They produce chemicals. The secretions from these gland cells are spread over the surface of the cnidarian by epidermal flagellae or cilia. Now remember, a flagella is a single long microtubule extension of the cytoskeleton. Cilia are many short microtubule extensions of the cytoskeleton. They move in a coordinated way to spread those glandular secretions over the surface of the animal, usually to do things like clean away dirt or to help lubricate the animal so they move through the water more smoothly. Now, there's a certain kind of gland cell found only within the anthozoans, and instead of producing a liquid secretion, it produces a secretion that immediately hardens. It's made out of calcium carbonate, and it's what makes up the stony part of corals. So that calcium carbonate skeleton, that matrix that corals build, is produced by these glandular cells. And then finally, you have stem cells. Stem cells have to be present so that they can replace epithelial cells or nematoblasts as they're used up. You want stem cells to be available so that they can constantly replenish your cell supply in this type of tissue. Now, this is an adult organism, not embryonic anymore. What kind of stem cells do you expect to see? Probably multipotent. They can produce any of a certain group of cells that all are part of the same system. So for the integument, that would be epithelial cells, nematoblasts, pigment cells, so on and so forth, all part of that related group of cells. Now remember, our cnidarians are hollow-bodied animals, by and large. When you have a jellyfish, they have their outer body covering, but then remember, they have that single opening that serves as both mouth and anus, and it opens into this hollow body cavity. The inside of this cavity is called the gastrovascular cavity. There we go. And the lining of this cavity has a special name. We call it the gastrodermis. Right? It's an internal layer of epidermis specialized for digestion and nutrient absorption. That's the function of the gastrodermis. It has a very similar structure to the epidermis, but with some slight modifications. This gastrodermis is made up of still epithelial cells. That's what the big pink cells are. But it's also heavily laden with gland cells. There's tons and tons of gland cells in your gastrodermis. There are also a lot of cilia. That makes sense, right? The products of those glands have to be moved around. They have to be spread out into that gastrovascular cavity so that those secretions, which usually are enzymes, those enzymes can be mixed with the food in the gastrovascular cavity and help to break it down. Then any nutrients that are released get absorbed back into those epithelial cells. Now remember, this is a diploblastic animal, right? So you have the gastrodermis on the inside, which is derived from the endoderm. The endoderm is what the gastrodermis started out as. Then you have the epidermis on the outside, which is derived from the ectoderm. But remember, there's that non-living layer of material in the middle. That manifests as the mesoglia in the adult animal. It's a non-living layer of cushion between the gastrodermis and the epidermis. Now, I'm going to erase all my drawings over here so you can see this box that just popped up. On this diagram, there's one structure we haven't talked about yet. It's this yellow thing. Notice this yellow thing shows up over and over again. There's one in every cell. What do you think it is? It's the nucleus. That's the nucleus for each of these cells. The periphery and the radiata have a fairly straightforward integument because they are very old lineages. They show us what the earliest forms of integument probably looked like. But when we move into our next clade, the bilateria, integument is going to get more complicated because this is the group of animals that really started to diversify, increase in size, and then move into new environments. We're going to start by talking about the invertebrate bilateria and then move on to the vertebrates where things get really complicated. Among the invertebrates, what you're going to see is the integument adapting to new environmental pressure and, in some cases, structural pressure that results from an increase in physical size. Exposure to harsher environments like open air, where desiccation is a serious risk, and also the need to support these increasingly large physical structures, is going to select for highly specialized integumentary systems, ones that are vastly more complicated than what we see in our soft-bodied radiata or porifera. However, the earliest bilaterally symmetric phyla, such as the platyhelminthes and the nematodes, they're going to act as a transitional group. Most of them are still marine or at the very least aquatic, so they don't have to contend with as many of the harsh factors that our later terrestrial groups will have to deal with. So they'll have a bit in common still with the radiata. However, they are going to start increasing in size and moving into new environments, even though they're still water-based. Those new environments are going to pose new risks, which our early bilaterally symmetric animals will have to adapt to. So their integument is going to start to be more complicated. We will start our discussion with the oldest bilaterally symmetric clade, which are the acoelomates. Platyhelminthes will be our representative acoelomate group. We are going to focus here on the effects that a new environment had on the evolution of their integument. Early flatworms were among the first to become truly parasitic animals, where they moved inside the bodies of other organisms and leached nutrients and resources off of them. That created a whole new set of physical stressors that these animals had to adapt to. No organism likes a parasite. They always try to fight against them. It's one of the things our immune systems are highly specialized for, is detecting and eliminating parasites. This means that a parasite is under constant attack from its host's immune system, and it has to have defenses that can deal with those attacks. Okay? It also means that the parasite has to locate a part of the body that's rich in resources, that's easy for it to access, and where it can anchor itself to live and feed, even though the host body is trying to dislodge it and get rid of it. Okay? There are non-parasitic platyhelminthes as well, however. They are going to act as our contrast, as our basis for comparison, to show just how unique and specialized parasitic integument for platyhelminthes has to be. Okay. All right. Remember that all of these adaptations to parasitism are forms of specialization. Some platyhelminthes have specialized on this new manner, this new lifestyle, and it has forced their integument to become more specialized as well. So let's start with a non-parasitic platyhelminthes so you can see what sort of the average integument looks like in this group. Then we'll move on and look at the parasitic platyhelminthes so you can see just how specialized they are. All right, class Turbularia, our planarians, are the perfect example of a non-parasitic platyhelminthes. Check out this cross-section you see at the bottom of the slide. This outer layer of yellow boxes, that's our integument. That's a layer of epidermis. Okay? It's made up of a single layer of ciliated epithelial cells. Notice again, our epidermis is only made up of a single layer of cells. That's going to be a common theme. You saw that the pinacoderm in sponges was one layer of cells. You saw that the epidermis in radiates, or in the radiata, was also just a single layer of cells. And here again, we've got an epithelial layer that is one cell thick. Right? Still made up of epithelial cells though. That's common to all of them. One unique thing, these cells may be syncytial, which hopefully you remember from the multicellularity lecture, means that they have more than one nucleus. They can be multi-nucleated. Now, all the epidermal cells here have cilia, but there are significantly more cilia on the ventral surface of the animal. Why? Think about how these animals move. They press themselves flat against a surface, and then seem to almost ooze forward. They're able to achieve that motion because all of these cilia down here, all of these hair-like structures, are pushing in the same direction at the same time. Those cilia are forcing the animal forwards, like 1,000 teeny tiny little legs. You also have sensory cells in the epidermis of these animals. Those sensory cells usually have elongated cilia. So the cilia stand out even further than normal, okay, sort of like this. And when those cilia are pushed over by a water current or because they're touched by another animal, they send an electrical impulse into the nerve they're attached to, which then goes to the nervous system of the animal and tells them that something in the environment has changed. There are gland cells as well, of course. The gland cells serve a variety of purposes. They can secrete a sticky mucus that helps with prey capture. That sticky mucus can also help them stick to their substrate. The substrate is the surface they're crawling across. Secretions can also help with motility. Motility means the ease of movement. They can act as a lubricant, which helps the animal slide across a surface or slip cleanly through the water without drag or friction. Finally, a very unique cell is present in this group. They're called rhabdite cells. The rhabdite cells are rod-shaped, and you can see them here. All right, there's a rhabdite cell. Here's another, and another. These rhabdite cells have a very special function. Their job is to produce a really nasty mucus. This mucus is incredibly distasteful, and it's secreted out onto the surface of the skin, where it spreads out thanks to those cilia that are moving glandular secretions across the surface of the animal. This is a predatory defense. When another animal tries to eat this flatworm, they're going to get a mouthful of this nasty mucus and spit the flatworm out. Flatworms can survive a lot of damage, as you'll see when we talk about reproduction in this group. One of the ways they can reproduce is actually by being fragmented, being broken into pieces. So even if that predator chows down on that flatworm and bites into them, the flatworm can often survive. The epidermis in this group is also the site of gas exchange. These flatworms absorb oxygen through their epidermis, and they also release CO2 back into the environment through their epidermis. They're able to do this because their surface area to volume ratio is very favorable. They've got a ton of surface area compared to their amount of volume because they're so small. This means it's really easy for them to absorb enough oxygen through their skin to provide that oxygen to all of the cells in their body. And it's also easy for them to get rid of all the CO2 that they're producing because they have so much skin through which it can be released. This last point is a lot of fun. In some species of turbularian, there are nematocysts, just like what you see in cnidarians, okay, in jellyfish and in hydrozoans. Nematocysts, in fact, in this group, are identical to the ones in cnidarians. And how is that possible? How could an identical structure evolve in two groups, all the way down to the DNA? It can't. Can't evolve in two separate groups to that degree of similarity. Can't be exactly the same. And yet these are. What that should tell you is that platyhelminthes, turbularians in particular, aren't making these nematocysts themselves. These nematocysts don't belong to these flatworms. Instead, the flatworms are stealing them. They're not made by the worm itself. Instead, these flatworms are consuming hydrozoans. hydras. They're digesting every part of the hydrozoan except for the nematocyst, and they're recycling those nematocysts into their own skin. The turbellarian takes the nematocysts from its digestive tract and moves them up to its own epidermis, where they function in the exact same way they did in their original host, which were the hydrozoans. Okay, so the turbellarians are stealing the nematocysts and then using them for themselves. All right, this is your baseline for comparison. These are our non-parasitic flatworms. Now let's take a look at how the integument of our parasitic flatworms differs from this. Remember that our parasitic classes of flatworm are class Cestoda and class Trematoda. Okay? Cestodes, again, are our tapeworms. Trematodes are our flukes. Both of these animals are endoparasites. Endo meaning inside. These are parasites that live in the insides of other organisms. This is compared to ectoparasites, ecto meaning outside, things like mosquitoes, things that parasitize the outside of your body. Fleas and ticks are other good examples of ectoparasites. Now these endoparasites have a whole range of hostile environmental conditions that they have to contend with, which means they're going to need a specialized and highly derived epidermis in order to protect themselves. The host's immune system and body are going to mount an intensely strong defense against these parasites to try to get rid of them. Okay? Likewise, the parasite, as it's fighting off the host's defenses, is going to have to make the best use of the time that it has by absorbing as many nutrients as it possibly can before the host's body kicks it out. To achieve these goals, parasitic platyhelminthes have a special type of integument that's just referred to as tegument. That's the name of this specialized form of epidermis. Okay. Now, once again, it's going to be one cell thick. Just like in our previous groups, the epidermis here, again, is one cell thick. But oh, those cells are very, very unique. All the cells, all the epithelial cells in this tegument are syncytial. They're all multinucleated. Okay? And only part of them is present on the surface of the skin. Okay? So this is one half of the cell, what I just boxed off here. That's one half of the cell. And this pink purple stuff, like what's in this box, that's muscle. Our epithelial cells are actually going to extend down into this muscular layer. So notice there's purple in between the cells here. There and there. Okay. That's all muscle. And our epithelial cells are extending down into it. Okay. Now none of these cells are ciliated. There's no cilia here. Okay. But these cells do have other jobs that they do. They protect against host defenses and digestive enzymes by absorbing that damage using all of this cytoplasm here at the top of the cell. The upper portion of that cell doesn't have any critical structures in it. The nucleus isn't up there, none of the organelles are up there except for a few ribosomes, maybe a few mitochondria. So that portion of the cell can take a lot of abuse without it harming the animal as a whole. That's the part of the integument that's going to be attacked by enzymes and acids and white blood cells. Okay. This upper portion of the integument is also very porous. You can see that on the plasma membrane up here where I just circled. There's a lot of gaps and holes. This is because the tegument is also specialized to absorb nutrients directly into the body of the parasite. Okay. Cestodes don't have digestive tracts because their tegumen is so good at absorbing nutrients, they don't need them. I'll show you what I mean. So that's the second point here. I'm going to skip ahead to it. Tapeworm tegumen has a bunch of little protrusions that you see here called microtrichs. Microtrichs are literally extensions of the plasma membrane of the cell. So instead of the surface of the cell looking smooth like this, it has all these finger-like protrusions on it. What this does is it increases the surface area of that cell. That increased surface area increases the total amount of nutrients that the cell can absorb. By covering their body with cells that have microtrichs, these tapeworms are able to absorb more than enough nutrients. And those are nutrients that have already been liberated from food. Tapeworms live in our intestines. By the time food hits your intestines, it's already being broken down and nutrients are already being released from it. That's the act of digestion. Food is pre-digested by the time it gets to the tapeworm. So the tapeworm doesn't have to do that digestive work. Instead, it can just absorb the nutrients that have already been released for it by the host's digestive system. It doesn't have to do any digestive work. It can just absorb those nutrients cleanly. For that reason, it doesn't need a digestive system of its own. Now, flukes have it a little bit rougher. Okay? They do have a digestive system because usually what they feed on is blood. Not all of them, but most of them. They absorb blood cells from their host and break them down. Okay? They have to anchor themselves in places where there's plenty of blood flow. Flukes love places like the liver because liver tissue receives a huge amount of circulation, whole lot of blood flow. Flukes anchor themselves in place using spines made of a protein called actin. Actin is a protein that's also found in chordate muscles. I tell you that so that you can see that some proteins, like actin, evolve over and over again in animals. They're really, really helpful configurations of proteins, and so when they show up, they get used again and again. Here are the actin spines on a fluke. Those spines embed in the tissue of the host and hold the fluke in place so it can't be washed away by the host's systems. Or at least it's harder to. Now, there's another parasite that we're going to discuss. It's not a flatworm, however. It's a nematode. Many, many, many nematodes are parasitic, but they have complex digestive tracts as well. There's no epidermal nutrient absorption in this group. That's a tapeworm thing, not a nematode thing. Now, nematodes have extra layers of defense and protection against their host's immune systems and against their host's digestive systems. Their outer epidermal layer is called the hypodermis. It's also syncytial, but it secretes an additional layer of protection in the form of a cuticle made of collagen. The cuticle's non-living. It's not made up of individual cells. It's just a thin layer of hardened collagen that acts as an extra physical defense for the nematode's body. Okay. The hypodermis also acts as a cushioning layer. It houses things like the nerve cords and also excretory ducts, which are used to get rid of waste. Okay. You can see on the diagram, top and bottom, There's what's listed as on the bottom, a ventral nerve cord. If you go up to the top, you see another little blue dot that is the dorsal nerve cord. The cuticle is the outermost layer, most superficial layer. If you look at the diagram on the right, it's that thick yellow layer on the bottom. And just in from that is the hypodermis, the living cells themselves. And you can see how the hypodermis wraps around the nerve cord there and provides cushioning for it. Now, the cuticle, its function is protection, but the trick is, since it's non-living, in order for the animal to grow, they have to get rid of their old cuticle and grow a new, larger one that they can then fill out and grow into. This is a process called ecdysis, or molting. It's a more common term for it. We're going to discuss how ecdysis actually happens at the beginning of part 2 of this lecture. But for now, know that nematodes are one of the groups who have to molt their non-living protective layer and regrow a new one every time they get larger in size. All right, that's it for part 1 of this lecture. I'll see you in part 2.